Hello everyone! This year MyOfrescent is hosting an inaugural Bioart event to present a new perspective in pathobiology and life science, including the complexities involving COVID-19 and its spread. This event will also discuss some stereological probes used in quantitative analysis. All of the paintings we are going to show today they represent micrographies or digital images which were obtained from an optical microscope, a fluorescent microscope, a scanning electron microscope, and also a CAT scan tomographer. Me and the artist who created the art painting, Miss Susan Barrow, we worked together for about 3 weeks in an attempt to combine the scientific information behind the micrographies or the digital images with the beauty of the abstract images to communicate its function to audience members who might not be from the biomedical field. And there are many hidden shapes of cells and the extracellular matrix components embedded in the images. And while the shapes may look abstract, in the reality, they are real and accurate depictions. All of these images they represent a dynamic motion of a biological reaction which was interrupted at some point. And the challenging situation here was to create a unique language where both scientists and the artist could see the same shape and science. So let's stop talking and take a look at this beautiful work. For example, this painting illustrates bovine pulmonar artery endothelial cells that have been labeled with Myrotreck red to identify mitochondria. It was also stained with Alexa fluor for actin filament and DAP for nuclei. Mitochondria are a very dynamic organelles and they generate most of the chemical energy needed to power the cell's biochemical reactions. At MyOfrescent, we count and measure these organelles using design-based stereology in order to be efficient. Design-based stereology is an accurate method of analysis where the measurements are as close as the true value. For this particular case, we estimated the fraction of the endothelial cells occupied by mitochondria and actin filaments. If you want more details of how this stereological probe works, please feel free to email us at info@myofrescent.com with the subject line counting mitochondria. The next picture is about a CAT scan of the chest of a young patient with COVID-19. He was an asymptomatic patient and the CAT scan was required to do it to a suspected lesion after an X-ray of the patient's chest. We made a stereological analysis of the patient's lungs and estimated the percentage of the lesions per volume of the lung for reference. This is part of a project in collaboration with FAPESP, a research institute in São Paulo, Brazil, and we are trying to create a database using stereological parameters to identify rare pulmonary lesions from COVID-19. The next picture represents a photomicrography of cardiac tissue which was immunostained to identify apoptotic cells. This is a classic apoptotic cells image which has a clover leaf-like nucleus. We counted the number of apoptotic cells and estimated the volume of the lesions in an experimental model of cardiac hypertrophy in rats. Last but not least, there are 2 scats of white blood cells as we observed via scanning electron microscopy. Here, the cells are in their living state, like this. This is a living cell, and the other one is an apoptotic cell infected with SARS-CoV-2 various particles here in red. Thank you for joining us today, and I hope you enjoyed the preview of the Bioart event planned for spring 2021. And if you have any questions about how quantitative analysis and stereology can be used in histopathology, please email us at info@myofrescent.com. Thanks. Bye-bye.